Thank you. Appreciate it.
All right, folks. Thanks for coming today. Despite the 9 minutes of technical difficulties, I think we're there. So today, I just want to introduce really quickly our speaker from within the department, postdoc Dr. Liza Loza. So Liza received her bachelor's from Bard College. I'm always going to screw this up. Jude Brooke or
Brooke Jude, yeah.
First name. With her, I screwed it up already. Went on to do her PhD with Tamara Doring over at WashU, where she spent a lot of time focusing on Cryptococcus and looking at the cell wall and how that contributes to the ability for the fungal cell to be able to go through its lifestyle. Just want to point out some different achievements that Dr. Loza has already walked through. F31 recipient during the graduate career. Had a really nice paper in PNES from their graduate research, another paper coming, and then has made some strong contributions already with some manuscripts in our line. So with that, pass it off to you.
Thank you so much, Matt, for the nice introduction. I don't have a microphone, but can everyone hear me okay? Okay, great. Okay, so I'm Liza. I'm really excited to tell you all about my work on molecular interactions between Candida albicans and the host. And I just want to say up front that I just started this project this year. So at the moment, I have a lot more ideas than finalized data. But I thought that giving this talk would be a really great opportunity to get insights and suggestions and feedback from this community. So really open to any of that you can provide. Okay, so what I'll be talking to you about today, first I'll give an introduction to the organism, then talk about studying adhesins in a non-model strain. Then I'm going to go into a bit of a deep dive / rant about one particular adhesin and contradictions about it in the literature. And then finally, I'm going to end on a really interesting aspect of the host response to Candida that we're excited to begin to understand. Okay, so Candida albicans is really unique among fungal pathogens because most of the time it's a normal member of our microbiome. So at least 50% of us are currently colonized with at least one strain of Candida albicans. It's really widespread, and it lives in the oral cavity, the gut, and the vaginal tract. And at each of these mucosal sites, Candida albicans cells form a really close association with epithelial cells. Changes in the host immune status or microbiome can lead to overproliferation of fungal cells and even invasion into the epithelial tissue by fungal hyphae. Oh, and this can kind of present as superficial infections like oral thrush or yeast infections. If this invasion isn't controlled, fungal cells can actually breach the bloodstream to cause disseminated disease. And this has about a 30% mortality rate in the US with proper antifungal treatment. So that's sort of the pathogenesis side of Candida albicans. This is a genetics audience. So I wanted to discuss the genetics a little bit. It's a diploid Ascomycete yeast with a genome of about 14 megabases, encoding about 6,000 genes. And this is a really important point. 70% of these genes are uncharacterized. So there's a lot of genetic space for us to explore in this organism. And within the species Candida albicans, different strains have up to 1% genetic variation between each other. So this is a really high level of genetic diversity. For comparison, within Homo sapiens, it's about 0.1%, so 10 times higher in Candida. And it does all of that, or it generates all this genetic diversity without even doing meiosis. And we really want to leverage this intrinsic genetic diversity to understand how it gives rise to phenotypic diversity. Because clinical strains of Candida albicans might exhibit more commensal-like behaviors or more pathogenic-like behaviors. They kind of exist on this spectrum. And I'm going to place two clinical isolates on our spectrum, SC534 and 529L. And SC534 is our reference strain. So almost all of the molecular work of the past several decades was done in this background. And it was isolated from the blood of a patient with systemic disease. 5290, on the other hand, was isolated from a patient with oral thrush, so still an infection, but not nearly as aggressive. And we can imagine that these different niches that they were isolated from might exert different selective pressures. If you put SC5, 3, and 4 in a mouse model of systemic disease by injecting it into their tail veins, it's uniformly fatal. So all the mice succumb within 2 weeks. 529L is sort of inconsistently virulent in this model. Only about 30% of the mice die. But 529L is really good at persistently colonizing epithelial sites. So in this experiment, they simultaneously inoculated the mouths and vaginas of 2 kinds of mice. And 5 weeks after infection, they're still able to detect 529L at both sites, but SC5, 3, and 4 is long gone. So I think of these 2 strains as the killer and the chiller. SC5, 3, and 4 is really good at killing mice in our systemic model of infection, and it's been really useful for studying certain virulence factors. 529L, on the other hand, is really good at just chilling with epithelial cells over time, which is in fact more representative of most Candida strains most of the time. But we've largely ignored the biology of this more commensal relationship because everyone has been studying the killer strain, which doesn't do that. And a classical microbiologist might say, well, of course you want to study the more virulent strain. Virulence is what we care about. But for Candida albicans, it's important to remember that every killer was once a chiller. So if we only focus on that end result, we're missing a lot of the basic biology of this organism, as well as possibly missing potential opportunities for therapeutic intervention to prevent this commensal pathogen transition. So hopefully I've convinced you why I don't want to study SC5, 3, and 4, but I instead want to learn how 529L sustains its persistent colonization of epithelial cells. So this is going to be my guiding principle or guiding question throughout the talk. And a previous grad student in the lab, Bobby, gave me a place to start. So he performed this RNA-seq experiment where he inoculated human oral epithelial cells with each of these strains so that we can compare their gene expression in this condition. And I'm particularly interested in a subset of genes encoding adhesins, which are proteins at the fungal cell surface that initiate or sustain physical contact with host cells via protein-protein interactions. So to be an adhesin, you have to be localized to the cell surface. You should have some kind of demonstrated adhesion effect on cells or substrate. And Candida encodes a big family of proteins that, because of their protein structure, are just sort of sticky to everything. These are pretty easy to pick out because they have this conserved protein structure, but it also encodes adhesins that interact in more specific ways with specific host proteins. And those are a little bit harder to figure out from sequence. And here it's also important to point out that we've only ever studied adhesins in SC5, 3, and 4, which, as you can see in the image on the left, is really good at making hyphae. And a consequence of that is that the best characterized adhesins are actually only expressed on the cell surface of hyphae. If you grow 529L in hyphae-inducing medium, the cells get a little bit elongated, but they're not making those true hyphae. And we know they're not expressing those hyphae-specific adhesins. And yet, we know it still has this close association with epithelial cells. So I hypothesized that 529L might be using a different repertoire of adhesins. So if we take a look at the differential gene expression data, I saw that SC5, 3, and 4 significantly upregulated 3 of those hyphae-specific adhesin genes. Basically, the usual suspects, these have already been well studied. On the other hand, 529L significantly upregulated 3 genes that were predicted to encode adhesins by a bioinformatics group back in 2011, but no one ever followed up to figure out if they are. So the big question is, are these actually adhesins? And if so, what are their substrates? So my primary approach is to delete each of these candidates from the 529L background using CRISPR, and then test their adhesion to epithelial cells and specific substrates. So the way this assay works is I put my mutants in with my oral epithelial cells, incubate them for an hour at 37 degrees, which is kind of the time point at which adhesion is happening. Wash them and then lyse the mammalian cells with the detergent, and then scrape each well and plate for CFU. And at the end of all that, I do not see any significant defects in adhesion to epithelial cells in my mutants. Interestingly, I see an increase in adhesion in mutant number 1, which we are curious about and trying to figure out if that's biologically meaningful or not. But since I didn't see any of these expected defects in my mutants, is this the end of the road for this project? I don't think so. And the reason is that Candida albicans has kind of made being sticky its whole personality. It encodes dozens of confirmed and predicted adhesins. So there's a lot of redundancy in this system. Additionally, some of these adhesins will adhere to more than one substrate. So this is just a really complicated milieu. So there's kind of two ways we can simplify this picture. One is to remove all the adhesins except for the one that we're interested in. So I'm doing that by individually expressing each of my candidates in Saccharomyces cerevisiae, which is less sticky than Candida albicans. And there I'm looking for a gain of adhesion in my transformants. Still working on the cloning for that. So I don't have any data to show you, but I think that that should be a useful approach. The other strategy we can take is to eliminate the whole complicated epithelial cell and just narrow in on one potential human protein that could be a substrate. How do we pick a substrate out of all of the possible human proteins that are made in epithelial cells? Luckily, there's a lot of literature about the kinds of proteins that Candida albicans likes to stick to, and many of them are extracellular matrix proteins. So the extracellular matrix consists of the basement membrane and the interstitial matrix, and its primary component is these long secreted proteins that kind of weave together and interact with each other and with epithelial cells, kind of forming a blanket over the epithelial cells almost. And from this previous literature, we know that Candida albicans cells stick to these four extracellular matrix proteins that are relevant to the oral epithelial model. So I'm basically just using these as a panel of potential adhesion substrates for my mutants. So this is still in progress. I can definitively tell you that none of these affect adhesion to fibronectin, but now I'm working my way down the list. So future directions for this part of the project is to test more of these substrates, obviously, to get the cerevisiae expression up and running. And then once we have both of these sort of approaches, we've got a plug and play system where we can put in as many adhesin or potential adhesins as we want. So an undergraduate in the lab, Megan Moran, is going to start looking at the other 60 predicted adhesins that were predicted by the bioinformatics group and prioritize which ones she wants to put into our systems. And the goal is to define the repertoire of adhesins used by 529L because we think that it's got a different lifestyle than SC534. This might be a more appropriate context to study a lot of the less studied predicted adhesins. Okay, so those are the putative adhesins. I neglected to tell you before that we also found significant upregulation of a known adhesin called INT1 in 529L. So there are several papers about this protein in the 80s, 90s, and 2000s. So we have a lot more background to work with. So how does it fit in with our adhesin criteria? It should be localized to the cell surface if it's an adhesin, but the evidence is really mixed. So this is the alpha-fold predicted structure for INT1. It's about 190 kilodaltons. It's got no signal peptide, no transmembrane domain, no GPI anchor. So we don't have any hints about its trafficking or localization from the protein sequence. It just looks like a soluble protein. And while early reports kind of assumed it was on the outside of the cell, this later paper actually tagged the protein with GFP, and we can pretty clearly see that it's at this bud neck and in fact is part of the septal ring. And they showed pretty convincingly that it actually physically interacts with septins in that ring structure. So how do we reconcile this with something that's on the outside of the cell interacting with extracellular human proteins? Um, there's a couple of ways I'm thinking about this. One is that, I don't know if you can see it up there, but in the 20-minute slide, you see a little bit of signal sort of at the edge of the cell and around the bud neck. So we think this cytokinesis and constriction of the bud neck might be a time that exposes intracellular proteins and like kind of could maybe push them outside even. So that might be one way. Another possibility is that there could be different isoforms of INT1. So this C-terminal tag this is like the full length protein. But what if there are truncated versions of this protein that may be extracellularly localized? And the same group did some engineered truncation, so they weren't replicating anything that we know of biologically. But I thought it was really interesting that when they truncated the N terminus, we see a lot more signal at the surface of the cell. So I'm looking into this both at the mRNA level and potentially at the protein level by incorporating a bunch of internal epitope tags into INT1 and seeing if any make it outside. Okay, probably the most important criteria is that we have some kind of adhesion phenotype. And this group saw that their INT1 deletion mutant had reduced adherence to HeLa cells compared to wild type. But they never figured out what human protein it's actually interacting with. So that's still a gap in our knowledge. And I'm going to show you this data from this paper. Adhesion is the y-axis, and then you can see the genotypes on the x-axis. And you can appreciate that the mutant, the third bar lacking both copies of INT1 has reduced adhesion compared to the first bar, which has 2 intact copies of INT1. Complementation of the mutant with an additional intact copy of INT1 didn't really rescue the phenotype. So it makes me a little bit suspicious about these strains. And I'm going to sort of hope you'll humor me as I deep dive into this strain background nightmare that occurred back in like the early 90s, or I guess early 2000s. Okay, so the way they generated these strains, this INT1 deletion strain, was kind of a convoluted set of steps, which was necessary at the time. Now we have CRISPR. It's really easy for us to delete both alleles of a gene at the same time, but they were constrained. So in this diagram, I'm showing you kind of the lineage tracing on the left, and then a green box indicates an intact and functional copy of the gene, and a red box is a non-functional copy of the gene. So of course, INT1 is the gene that we're interested in. They wanted to delete both copies. URA3 is an auxotrophic marker that's probably familiar to the yeast people here. So this was a way to make their transformant selectable because mutants lacking both functional copies of URA3 can't grow on uracil and uridine, or can't grow on medium lacking uracil and uridine. Whoops. Okay. Unfortunately, URA3 is right next to a transcription factor called IRO1. So when they deleted each copy of URA3, they accidentally truncated IRO1 as well. So that means that in the INT1 deletion mutant, the full INT1 deletion mutant, they've got no functional copies of IRO1, and yet they're comparing in all of their studies, their wild type control strain is this CAF2, which still has a functional copy of IRO1. So it's not the ideal background control. Does this even matter, right? I mean, who knows about IRO1? Maybe it doesn't matter to anything we care about. It turns out that it matters to at least one phenotype we care about, which is filamentation. So filamentation is the ability to grow hyphae, and we can kind of simulate this in the lab by growing strains on specific medium that induces that filamentation response. So in this paper, they claimed that their INT1 deletion mutant had a loss of filamentation based on these images. So you can see that the wild type it actually has like differences from the real wild type, sort of has these like wrinkly star colony morphologies. but INT1 sort of looks like classic yeast colonies. When I made my INT1 deletion mutant, I also wanted to test this phenotype and see the loss of filamentation, but hopefully you can appreciate both of these strains filament just fine. And actually, there's a little bit of a colony morphology difference in my filamenting strains versus the CAF2. So let's keep that in mind. Okay, so I hypothesized that maybe it's all these background mutations that are actually contributing to this phenotype rather than INT1. So what I decided to do is kind of take a sledgehammer to my strains and delete both copies of UR3 and truncate both copies of IRO1. And what I see is a growth defect, which is probably from the double UR3 deletion, as well as a filamentation sort of change. If we zoom in on these colonies, I don't know, they're a little bit blurry, but now we see this like wrinkly star phenotype. So I think that that's actually probably attributable to IRO1 rather than INT1, because INT1 doesn't seem to make a difference in this phenotype at all. So I'm probably not going to make like every permutation of these strain lineages, but my plan is to at least make an IRO1 deletion mutant to show that that is probably driving this reported filamentation defect in INT1. And I should say that this work was done with the help of Amanda Coleman, a genetics graduate student while she was working in our lab. Okay, so we looked at adherence to cells. We think that that's true, even though the backgrounds are a little bit iffy. We still don't know what substrate it binds to. So I, of course, put my INT1 deletion mutant into the epithelial cell adhesion assay. Unlike with their experiment with the HeLa cells, I don't see any significant difference between it and 529L. But I had a little bit more luck narrowing down a possible substrate for INT1. And that's because in 2008, some researchers looking at a Candida albicans ortholog in a distantly related fungus, Pneumocystis girovecii, looked into what its adhesion specificity was. So their organism wasn't genetically tractable at the time. So instead, they expressed their INT1 homolog in Saccharomyces cerevisiae. So again, now we're looking for gain of adhesion rather than loss of adhesion. And it's pretty compelling that they see sort of a specific adhesion to fibronectin, but not to other extracellular matrix proteins or a control protein BSA. So this was a great hint for me, so I decided to test this mutant with fibronectin. And indeed, we do see that the INT1 deletion mutant has about a 30% reduction in adhesion to fibronectin compared to 529L. And this defect can be rescued by complementation of the mutant with either allele. Okay, so this suggests that INT1, like its distant cousin in Pneumocystis, binds to fibronectin, solving a long-standing mystery about this protein in Candida. Okay, so future directions for this part of the talk, I mentioned the IRO1 deletion, and then I'm also investigating the possibility of these different potential INT1 isoforms, and if I can, trying to understand how INT1 gains access to the extracellular space. And this is to reconcile contradictions in the INT1 literature. Okay, so coming back to my guiding question, how does 529L sustain its persistent colonization of epithelial cells? So up till now, I've been focusing on these fungal adhesins, everything on the fungal side. But in fact, this is a two-way interaction all the time. They're responding to each other. So what about what's happening in the epithelial cells? And in a previous study in the lab, we infected the oral cavities of mice with each of these strains, and we saw really different immune responses. So in the SC5, 3, and 4 infected animals, we saw high levels of these pro-inflammatory cytokines, whereas 529L infected tissue was not inflamed, either in this assay or by histology as well. So is this just a matter of SC5, 3, and 4 hyphae just punching through and doing a lot of damage, so we're getting all of this inflammatory response? Or is it possible that 529L could be inducing a state of immune tolerance? And both of these things can be true at the same time. And again, we've only studied that SC5 through and 4 side of the equation and not the 529L end of the spectrum. So to start thinking about this, we returned to the RNA-seq experiment, which was actually a time course. And instead of looking at the fungal genes expressed differentially between the strains, now I'm looking at the host genes that are expressed differentially when they're exposed to each of these strains. And we further narrowed it down to, or we further filtered our list by looking for genes with possible immunomodulatory effects. And that's how we got to this pair of genes, whose ITGAV and ITGB6. And their products dimerize to form the integrin alpha V beta 6. And hopefully you can appreciate that the presence of 529L, but not SC5 through and 4, leads to increased expression of each of these integrin subunit genes over time. Now, to explain why we're excited about this result, I have to take a step back and explain a little bit about what integrins are. So these are heterodimeric membrane-spanning receptors at the cell surface of many cell types, including epithelial cells. And they interact intracellularly with the cytoskeleton, but also extracellularly with various ligands. And they're capable of transmitting both inside-out signaling and outside-in signaling. There are 24 integrin subunits in humans that dimerize to form 18 heterodimers, 18 integrins. And integrins just do a lot of really complicated biology, and they do a lot of like disparate things. So I'm going to focus our attention back again on the one we're interested in, which is alpha V beta 6. Okay, so this integrin is expressed at really, really low levels in epithelia. It's an RGD binding integrin, so it binds proteins via their RGD motifs, which is pretty common for integrins actually. But what's really unique about this integrin is that it activates the cytokine transforming growth factor beta 1. I'm just going to call it TGF beta 1. And this partnership has been implicated in processes like wound healing, as well as immune tolerance of commensals. So that really struck our interest that this might be something relevant to Candida albicans, but particularly 529L, which has this persistent colonization phenotype. Okay, so what does this partnership look like? Latent TGF beta 1 propeptide is secreted from the cells out into the extracellular matrix. And its RGD motif is bound by alpha V beta 6. And that leads to the release of active TGF beta 1. It's kind of released from its cage. And locally, this creates a positive feedback loop. So more active TGF beta 1 increases the expression of the alpha V beta 6 subunit genes, which leads to more active TGF beta 1, which might account for the effect that we see of sort of growth in expression over time. Okay. So I mentioned that TGF beta 1 is a cytokine. Cytokines have really interesting and wide-ranging roles. What does this one do? So broadly speaking, the downstream effects of TGF beta 1 are immunosuppressive. And they do that by suppressing the proliferation and differentiation of T cells. And this can happen sort of at different sites in the body, but it does happen locally, pardon me, with like peripheral T cells in epithelia that we'd expect to be patrolling epithelia. Okay. So that kind of effect wouldn't be happening in our little dish of oral epithelial cells, but we think that it might be relevant in our mouse models, and it might explain why we see such a less inflamed environment, even though 529L is robustly colonizing the oral cavity of mice. Okay, and the other thing it does is to increase extracellular matrix production. And as you may remember, extracellular matrix proteins are really where Candida albicans likes to hang out. So this is very speculative, but we think that it's possible that this might create the extracellular, increased extracellular matrix protein production, might be making a nice little extracellular nest for these candida to exist in. This is kind of their preferred substrate. And we have some evidence that this might be the case, although we're interested in soliciting ideas for other ways to test this. We see that antibody blockade of alpha V beta 6 or siRNA knockdown of either integrin subunit leads to increased 529L invasion or internalization into epithelial cells. So the idea here is that, 529L prefers to just hang out on the porch, but if the porch is not hospitable for whatever reason, it can go deeper into the cells. But maybe this is a mutually beneficial interaction where epithelial cells kind of keep the colonizing strain away from the actual cells, and 529L gets to exist in its little happy nest of extracellular matrix proteins. Okay, but how is it doing this? How does 529L induce this upregulation of the alpha V beta 6 subunit genes? So this is sort of a proposal part. This is what I'm planning to do. I'm planning to use this increased expression of these two subunits as a readout, which I'll be testing by RT-qPCR rather than RNA-seq. And I'm planning sort of two orthogonal approaches. So one is classic cell biology. I'm going to test whether heat-killed or fixed cells can still trigger this upregulation of these two integrin subunit genes. If they can't, that kind of suggests that it's an active cellular process. And then I can go from there. I can do cellular fractions to look kind of narrow down what might be the effector here. So for example, if the supernatant can induce this upregulation of expression, we would guess that it's something that's secreted, right? And then we can protease treat to make sure, see if it's a protein or not, and so on and so forth. So we're hoping to get at least a better idea of the characteristics of whatever it is that's driving this upregulation of these two genes. Okay, the second approach is quantitative trait loci, or QTL mapping. So in this strategy, you start with two strains with opposing phenotypes, which of course we have, SC534 and 529L. Previously in the lab, we have mated these two strains and generated parasexual progeny with known recombinant genotypes or defined recombinant genotypes. So then with all of these recombinant progeny, I would do this assay. It's a quantitative assay that would result in sort of quantitative variables that we could then map the loci to causative alleles via this method. Okay, so this is just basically another way of saying what I just said. These purple progeny, they're going to have varying amounts of SC534 versus 529L DNA in them. And we can use that to look for genetic differences that are driving this phenotype. And we may find those in the actual effector itself, or it may be a factor upstream. Okay, so future directions for this, I'm using these two approaches to try to figure out the 529L factors that drive this upregulation of ITGA-V and ITGB6. And then, as I mentioned, I'm interested in doing mouse studies to look at the potential immunosuppressive effects that are downstream of TGF-beta1. And we're really excited about this. First, because alpha V beta 6 has never been implicated in any kind of candida interaction before, but also because it's so strain specific and it seems to really only happen with our persistent colonizing strain. So we think that this might be a way of sort of creating this environment of immune tolerance where Candida albicans can exist long term. Okay. So to summarize, We're really interested in leveraging the genetic diversity of Candida albicans, and especially these non-model strains. If 70% of the genes in our reference strain are uncharacterized, I think there's even more unknown biology in our non-model strains to look at. So I'm looking at this interaction from both the fungal and the host side. So I told you about investigating predicted adhesins in a non-model strain, and showed you that INT1 adheres specifically to fibronectin, which is a new finding for the field. And then on the host side, we see that this integrin has this really strain-specific response that we are interested in dissecting, both how it's induced and then also the downstream sequelae. Okay, so with that, I'd like to thank you all for your attention. Thank the entire Anderson Lab for all their support, our collaborators at UCLA and here, and our funding sources. And I'm happy to take any questions. Yeah. Ooh, that I have never seen anybody, at least for Candida. I don't know if that's true, but that's a really interesting idea. Yeah. Yeah, that would be really cool. That's like a very testable one to try. Yeah, but thank you. That's interesting. Mhm. Yes, that's a good question. So the paper that tagged it, they were looking, I think they were expecting to see that. And they saw, the only thing that they saw was sort of a delayed recruitment of a septin that interacts with it, but it actually didn't change sort of the overall process of cytokinesis. Go ahead. Yeah. To prioritize. Yeah, I'm definitely not going to ask her to knock out all 60 genes. Um, I am planning to just, the problem is most of them are uncharacterized still. Like we just have no idea what they do. They did in this bioinformatics paper, they kind of gave it like a high or low predictive score. So I'd imagine we'd probably be picking ones that they're more sure would be adhesins. Um, yeah, I think we can just kind of do as many as we can do and maybe even just phenotype them like with other assays unrelated to adhesion, just to sort of get a little bit, further into the black box of these uncharacterized genes. Mhm.
There are other strains of hand outcomes that are persistent colonizers.
Yes.
Have you or are you planning to look at how those strains perhaps impact this off beta-6 expression?
Yes, that is something I am planning to do. If I had to make a prediction, I'd say I think that the persistently colonizing strains we'd see the same sort of upregulation. I don't know that for sure, but definitely something to test. Mm-hmm.
That are differentially expressed or regulated that can repurpose proteins or remove splicing?
Yeah, so Canada albicans does a lot of, I think they call it like non-canonical secretion. So for example, like um, mm, there's an alcohol dehydrogenase that should be intracellular, and it is intracellular. It's in the cytosol, but it's also an adhesin because it somehow ends up on the outside of the cell despite having none of the usual sort of signals that we would expect to traffic it there. So I think that there is precedent in that for other proteins and even adhesins in Candida albicans. What was the second part of your question?
What is contributing to that? and seen any changes,
Mm.
hints about what could be causing that to happen?
Yeah, I think I don't really know. I do think we know that like during cytokinesis and like the bud neck sort of constricting, we do see like exposure of different glycans that are usually underneath the other glycans of the cell wall. So I think like that would sort of be my a guess of like how that could be happening is maybe things that are everything is getting compressed into a small amount of space and maybe things are like the wall is a little bit different and it's a little weaker, and maybe things are able to get out sort of at that kind of pressure point. But yeah, I have no proof for that. This is all very speculative. All right. Yeah, if there's no last minute questions. I would just want to thank you all for the opportunity to kind of introduce myself to you and open to collaboration and feedback and suggestions in the future. So thanks.